Matthew Galsky. He's a very well-known medical oncologist from Mount Sinai in New York, and he will talk about muscle-invasive bladder cancer. We heard a little bit actually in the last talk, and he will tell us about the immunotherapy, what are we doing now, and what will we maybe do in the future. Thank you.
Thank you. Thanks to the organizers for having me. Here are my disclosures. There was a recent study that showed that disclosure slides were shown so quickly that they were too quick to be viewed by the human eye. At the same time, I don't know if I'm supposed to stand here and wait for you to read it all. So I'll move on. So it's been challenging to develop new treatments in advanced bladder cancer. And this slide shows FDA approvals for new drugs in bladder cancer over a few decades. And you can see from 1995 to 2015, there was a single drug approved by the US FDA, that was valrubicin. So a long time without progress. And then in 2016, we have 2 immune checkpoint inhibitors approved, and then 3 more in 2017. And so certainly a change in the landscape of the treatment. These are the data sets that led to the approval of these drugs by the US FDA. Most of these were single-arm studies with response rate endpoints, accelerated approvals. And here I've plotted the size of the arm of the study and the response rate. And what you can see here is that these drugs were not approved because they work in everyone. Unfortunately, that's quite the opposite. That the response rates with the PD-1 and PD-L1 inhibitors in platinum-resistant patients with metastatic urothelial cancer range from about 15 to 25%. The enthusiasm for this class of therapies, as most of you know, has been more based on what happens when patients do respond. So this is one of my patients that I met in 2014, a retired physician who had metastatic urothelial cancer to the pelvic and retroperitoneal lymph nodes. And he initially was treated elsewhere with gemcitabine and carboplatin, ended up with neutropenic sepsis, actually had fungemia, was in the ICU, went into renal failure, eventually recovered, by that time had progression of disease, was enrolled on one of the early studies with PD-L1 blockade, and you can see his scan a few months ago with a complete response that's maintained while receiving therapy. I think it's cycle 70 or something recently. So this is what can happen, and this is why there's enthusiasm about these drugs. The other reason that there's enthusiasm is that for the majority of patients, and notwithstanding bad things that can happen, and it would take another full lecture to discuss immune-related adverse events and the management in the spectrum of what can happen. But notwithstanding some bad things, the majority of patients tolerate these treatments quite well. And anyone who has been involved with any of the studies in the adjuvant setting or any other studies that randomize patients to immune checkpoint blockade versus placebo, these double-blind randomized studies, that's, I think, the best testament to the tolerability of these drugs, because when we run these trials, I cannot tell who's on immune checkpoint blockade versus placebo for the most part, nor can patients. And it would be hard to say that about any of our other systemic treatments. So grade 3 and 4 adverse events occur in about 7 to 15% of patients. These are the rates from the trials that led to the approval of these drugs. Confidence intervals are overlapping, so I'm not showing this to suggest that there's a difference between therapies, but they all cluster pretty well together. These drugs have been well tolerated enough that they've been extended to populations of patients that historically have been difficult to treat. And that includes patients who we've considered cisplatin-ineligible, patients with comorbidities that render the risks of cisplatin-based chemotherapy higher than the potential benefits. These are usually older patients with several comorbidities. And so single-arm trials have been run in the chemotherapy-naive population of patients who are ineligible to receive cisplatin-based chemotherapy, again showing response rates of about 24–25%, leading to the approval in the United States of both of these therapies as first-line treatment in this unmet need population. So we have a class of therapies, and what I didn't mention is that looking across these therapies, they look pretty similar to me. We'll probably never have randomized comparisons of these drugs, but the response rates across data sets are pretty similar. So we have a class of therapies that work really well in a small subset of patients, and of course that raises the issue, can we select better who should receive these treatments? The predictive biomarker that's been explored most extensively to date has been expression of the ligand for PD-1, PD-L1 by immunohistochemistry. And as many of you know, this field has been a little bit of a mess. And it's been a little bit of a mess because with every therapeutic antibody that's being developed, there's a different diagnostic biomarker that's being developed. And not only is there a different diagnostic biomarker, but the assay, the way that it's scored is different. So sometimes just looking at tumor cells, sometimes just looking at immune cells, sometimes looking at both. So a lack of harmonization here. And we know that there are differences between these antibodies. I'm showing data in lung cancer here. Something similar has been done in bladder cancer. This is called the Blueprint Project, led by Fred Hirsch. So these are individual patients with lung cancer. They're tumor specimens. Each specimen is stained with all 4 of the antibodies for PD-L1. This is looking at tumor cell staining, here looking at immune cell staining, and you can see individual tumors where the scoring with the different antibodies is completely disparate, which could potentially render one patient by one assay to be eligible for treatment versus the other assay. And this is probably more of an academic discussion, probably more than just an academic discussion, in that failure to completely appreciate the performance of biomarkers can have consequences in terms of clinical trials design and interpretation. So I mentioned that we have we have most of the approvals for immune checkpoint inhibitors based on single-arm trials with response rate endpoints, but we actually do have 2 reported randomized phase 3 studies now. One of them with pembrolizumab, KEYNOTE-45, and another with atezolizumab, IMvigor211. These studies were done in patients with metastatic urothelial cancer who had progressed on platinum-based chemotherapy. The comparator arm was the same, standard of care chemotherapy, which included taxanes or vinflunine in countries where vinflunine was available. Each study included prospective testing for PD-L1 and stratification based on PD-L1 expression using the assay that's been developed along with that therapeutic antibody. Both studies used overall survival as a primary endpoint, and both studies prospectively defined looking at overall survival in the entire population and in the PD-L1 high defined population. But the way that this was done between the 2 studies differed. In KEYNOTE-45, the alpha was split so that both of the analyses could occur simultaneously, overall population and the PD-L1 high population. Whereas in the IMvigor study, a hierarchical statistical analysis plan was used so that the PD-L1 high population had to be positive to move on to the overall population. And this is what happened. Here's the results with the Keynote study. In the all-comer population, there was a survival benefit along with the PD-L1 high population. But in the IMvigor211 study, here's the hazard ratio in the PD-L1 high population, which did not reach statistical significance. Therefore, the all-comer population analysis was simply exploratory, did suggest a benefit. Why did this happen? Well, in part, likely because of the performance of the biomarker. And you can see here in KEYNOTE-45, the outcomes, and I'm showing response rate here, with pembrolizumab versus chemotherapy in the all-comer population and the PD-L1 high population. And you can see that the response rate's higher in the PD-L1 high population with pembro, and the delta changes a little bit in favor of pembro in the PD-L1 high population, suggesting perhaps some predictive performance. But look at what happens in the IMvigor211 study. Here's the all-comer population, really no difference. And in the PD-L1 high population, both groups had higher response rates, suggesting that PD-L1 expression based on this assay might have been more prognostic than predictive. So this has consequences. Now, to make matters even more confusing, and don't ask me to explain this because I can't explain it yet, based on 2 large ongoing studies in the first-line metastatic setting that are poised to change the treatment paradigm in metastatic urothelial cancer. Both of these studies are randomizing patients to receive immune checkpoint blockade alone versus chemotherapy alone versus the combination of chemotherapy and immune checkpoint blockade. Both of these studies, based on the Data Safety Monitoring Board's recommendations late in accrual, had stated that patients who were PD-L1 expression low should no longer be randomized to the single-agent immune checkpoint blockade arm. The FDA reviewed that data, even though this has not been analyzed. The results of this trial have not been released. It actually was ongoing at the time of this event. The FDA and other regulatory authorities reviewed the data and changed the label to restrict the use of immune checkpoint blockade in cisplatin-ineligible patients to PD-L1 high expressing tumors. So despite the disparate nature of these assays, for some reason in these studies, they're performing similarly in suggesting that patients who are PD-L1 low should not get immune checkpoint blockade as a single agent. So these trials, while not yet finished, not yet reported, have already changed practice, and certainly everyone's anxiously awaiting review of the full results of these trials. So this is what the treatment paradigm looks like today. For the first-line treatment of patients with metastatic urothelial cancer, we really separate patients into those that are cisplatin eligible. They're still getting cisplatin-based chemotherapy. Cisplatin ineligible but PD-L1 high, immune checkpoint blockade. Cisplatin ineligible, PD-L1 low, carboplatin-based chemotherapy. Patients who are, quote, chemotherapy ineligible, and this has come up because patients enrolled on the single-arm trials of first-line immune checkpoint blockade had characteristics that could suggest an excess risk with chemotherapy. And so this is sort of a newly defined category, and these patients can receive per label in the United States immune checkpoint blockade. And then the second-line setting, based on what was received in the first-line setting, of course, that dictates what's administered. So that's the past and the recent present. The future, at least the near future of immune checkpoint blockade, can really be broken down into 3 major categories. One is moving these drugs into earlier disease states, and I'll touch on that a little bit. 2 is combinations. These drugs work in a small subset of patients. Can we extend the reach with combination therapy? And then the 3rd is biomarkers. So we all know that metastatic urothelial cancer accounts for a very small subset of patients with bladder cancer or urothelial cancer, and certainly the prevalence and incidence of patients with non-muscle invasive and muscle invasive disease is much higher. So the potential for impact with new therapies is certainly higher. And perhaps one of the largest areas for impact with systemic therapy is in the muscle invasive disease setting. This is certainly a window of opportunity for potential cure with better systemic approaches, because we know, and this is the data from the SWOG study, we know that even with neoadjuvant chemotherapy and cystectomy, a large proportion of patients will develop metastatic recurrence and die. So we certainly have a lot of room to improve the treatment of this disease state. Alex alluded to 2 trials that have been done, 2 moderate-sized single-arm phase 2 studies of immune checkpoint blockade prior to cystectomy. The Abacus study, run by Tom Powles, administered just 2 doses of atezolizumab prior to cystectomy. This was for patients who were deemed cisplatin ineligible. The PURE study, led by Andrea Necchi, included 3 cycles of pembrolizumab prior to cystectomy. The patient population was slightly different. This was open to all comers, not only cisplatin ineligible patients. There was some restriction on the T stage of the tumors, but relatively similar patient populations overall. The primary endpoint of both of these studies was the complete pathologic response rate. And here you can see the complete pathologic response rate from the Abacus study and the PURE study, and I've plotted this relative to the complete pathologic response rates from other neoadjuvant cohorts. And of course, with any neoadjuvant study, with any single-arm neoadjuvant study, we always have to remember that there is a background pathologic complete response rate with TUR alone, with no systemic treatment. That's about 13% in SWOG 8710. And so you can see that with immune checkpoint blockade, there does seem to be a signal that's raising above this background, and certainly a lot of enthusiasm for these drugs in this disease state based on these two trials. And now there are about 6 large international randomized studies being done in the muscle-invasive disease setting, randomizing patients to immune checkpoint blockade alone, immune checkpoint blockade followed by cystectomy versus cystectomy alone, and then another group of studies randomizing patients to chemotherapy plus immune checkpoint blockade versus cystectomy. Those studies are just getting started, so it will be several years before we have those results. Alex also alluded to Keynote 57, so I'm just going to say one word about this. Of course, this was done in BCG-unresponsive patients. There were 2 cohorts of patients enrolled. Only the carcinoma in situ cohort has been reported to date at ESMO. And you can see here that the 3-month CR rate was 38%, but look at the patients remaining in CR at 9 months. I have to say I'm a bit disappointed with these results, and I think that demonstrating any activity with immune checkpoint blockade in this population is important because we didn't know this before this trial, that these drugs would be active in this disease state. But certainly systemically administered treatments with the risk for serious side effects, including treatment-related death seen on this study, I think we have to really think about this. And there have been a large number of studies launched, large international randomized phase 3 studies in this disease state, and I think we need to be a little bit thoughtful about how rapidly we apply these systemic treatments to this early curative population. I'm going to talk a little bit about combinations, and certainly that's what we do as medical oncologists. We combine things. I think that we often like to think we're combining things because of some understanding of mechanism, some understanding of resistance, some appreciation that synergy might be achieved. But in fact, we know very little about why combination therapies work in oncology, including the combinations that we use today. And really, combination therapies in a patient population could work because the drugs are synergistic. That's what we hope for. They can work because they're additive, or they could potentially work because of this principle of independent activity. And this study from Palmer and colleagues in Cell in 2017, I think was one of the best studies I read in 2017. I think really sheds light on how little we know about this, but how important this is in terms of how we think about drug development and biomarker development. So what they did in this study was take data from clinical trials and data from PDX models, and they modeled what the likelihood that these combinations would be additive, synergistic, or just result from independent drug action. That is that in a population of patients with heterogeneous tumors, does the combination of 2 drugs work only because you're increasing the likelihood that a patient's going to respond to one of the drugs? They might be responsive to one, they might be responsive to the other. There might be some overlap. By giving 2 drugs, you're just hedging your bets. And what they showed here, that the majority of combinations that we use now are probably a result of independent drug action, not additivity or synergy. That's based on modeling data, but a very well done study. And it really does make you think a little bit about what we're trying to achieve with combinations at the very fundamental level. And if you're the CEO of a drug company, this is important data, because do you tell your teams that they should seek out the drugs with the highest single agent activity and combine them together? Or do you really think that probing into biology and mechanisms of resistance is the most important thing in developing rationally designed combinations? I don't think we know the answer to this, and both approaches are being pursued, and that's probably the right thing to do for now. The two combinations that have been explored most extensively to date in urothelial cancer are combinations of PD-1 and PD-L1 blockade with cytotoxic chemotherapy and combinations with CTLA-4 blockade, like in other solid tumors. Here's the data available to date with PD-1 blockade or PD-L1 blockade plus CTLA-4 blockade. And you can see that fairly large single-arm data sets, there does seem to be some increase in activity with combination therapy as opposed to the 15 to 25% that we see with single agents. There does seem to be some dose-response effect with CTLA-4 blockade that isn't necessarily seen with PD-1 or PD-L1 blockade. That's not just in bladder cancer. This has been shown in melanoma and other cancers as well. And so getting the dose of CTLA-4 blockade is probably very important and has been underappreciated. There's actually only been one study of CTLA-4 blockade as a single agent in metastatic urothelial cancer, and it's flown under the radar a little bit. It was presented at SITC in 2018, smallish 32-patient study, but an 18% response rate with single agent CTLA-4 blockade. Not great, but not that much different than we see with PD-1 and PD-L1 blockade. So this drug class does have independent activity in urothelial cancer. We'll find out if the combinations are beneficial relatively soon. The Danube study was the first major phase 3 study in patients with metastatic urothelial cancer in the frontline setting, integrating immune checkpoint blockade to be completed. The results will probably be available sometime this year. And this randomized patients to single agent PD-L1 blockade versus the combination versus chemo alone. Checkmate 901 is further behind, still accruing and randomizing patients to ipi-nivo versus chemo. And there is an arm for cisplatin-eligible patients who will also be receiving gemcitabine, cisplatin, and nivolumab. There's not a direct comparison between these three arms. Rather, there's not a direct comparison between these two arms, but this is the only trial ongoing where these different treatments are being administered within the context of the same trial. So at the end of the day, if we have doublet IO therapy and chemo plus IO therapies, how do we choose which one? This will be the only data set where we might be able to glean some insights into that. There are multiple reasons for potentially combining chemotherapy with immune checkpoint blockade. These are the reasons that are discussed most frequently. The potential to cause immunogenic cell death. This is a very certain, very specific type of cell death. Release of danger signals, which results in recruitment and maturation of dendritic cells. Not all cytotoxic drugs do this in the laboratory. In fact, cisplatin and carboplatin reproducibly do not induce immunogenic cell death in the laboratory. Oxaliplatin does, interestingly enough. We have tried to document immunogenic cell death in patients in clinical trials. It's been a very difficult thing to do because we don't know exactly the right way to test for this in terms of assays. We don't know the right timing to procure biospecimens to interrogate for immunogenic cell death. But based on some very simplistic assays, we've not been able to show that this happens in patients, and there isn't a lot of definitive evidence of this occurring in patients, but as the ability to collect fresh tissue and with some of the more high-dimensional approaches like single-cell-seq that are available today, perhaps we will be able to document this. We do have a trial right now randomizing patients between PD-1 blockade plus gemcitabine and carboplatin versus PD-1 blockade plus gemcitabine and oxaliplatin to test whether or not the platinum drug really does make a difference. Depletion of immunosuppressive cellular subsets is discussed a lot. Depletion of myeloid-derived suppressor cells, or Tregs, also very little evidence that this happens in patients. And of course, this concept of non-cross resistance that I mentioned. We know chemotherapy works in some patients. We know immune checkpoint blockade works in some patients. The combination might increase the likelihood that patients are going to respond to something. So a bunch of randomized phase 3 trials are being done to test this concept, and Keynote 361 and Vigor 130, which I already mentioned, have completed accrual, and we should have some initial results from these trials within the next 18 months. NILE is just getting started. NILE is the only study to test this really kitchen sink approach, which is combining chemo and CTLA-4 blockade and PD-L1 blockade. So if we really think that understanding mechanisms of sensitivity and resistance are important, then understanding biomarkers in the clinic will be critical to developing rational combination strategies. And most of the biomarkers explored to date have really focused on our understanding of the mechanism of action of these drugs. That is, that to respond to PD-1 or PD-L1 blockade requires a pre-existing anti-tumor immune response, which is subsequently restrained by the tumor microenvironment because of the release of interferon gamma and the then upregulation of PD-L1 in the microenvironment. If this is true, then measures of adaptive immune resistance should predict response to immune checkpoint blockade. That is, things like PD-L1, which we already discussed, CD8 infiltration, interferon gamma gene signatures, and it turns out that all of these do correlate with response to immune checkpoint blockade, and it's really this constellation that defines hot tumors versus cold tumors. These biomarkers, which again have all been correlated with a higher likelihood of response to immune checkpoint blockade, are also highly correlated with one another. So this probably represents one category of biomarkers, these inflamed microenvironment biomarkers. The other class of biomarkers that's been explored fairly extensively is, of course, tumor mutational burden, which is felt to be potentially a surrogate for the presence of antigens or neoantigens. There have been several data sets looking at this. Here's from the Checkmate 275 study, where just looking at tumor mutational burden cut at the tertiles allows one to distinguish higher versus lower response rates and higher versus lower survival. The appropriate cut points in urothelial cancer have not been defined. Recent data suggests that the cut points for TMB probably will have to be tumor type specific. One of the interesting things about TMB, though, and this has now been shown by several different groups, and I don't have a good explanation for it, is that TMB doesn't correlate with these biomarkers. You would think that if this is a surrogate for antigen burden, then higher TMB would be associated with higher T-cell infiltration, interferon gamma, et cetera. But it turns out that these correlate very weakly, and in fact, they're independent predictive biomarkers. So why this isn't clear, and I'm not sure we actually have a good understanding of what TMB is telling us. Like a lot of groups, we've been interested in understanding what drives cold versus hot tumors as a way to define potential novel predictive biomarkers and mechanisms of resistance. The way that we do this is, like a lot of other groups, we use available gene expression data to infer the infiltration of T cells based on T cell related gene sets, and then look for correlations between those gene sets and other pathways, or lack of correlation, that is, what is correlated with a lack of T-cell infiltration. When we did this initially, we were surprised to find a very high correlation between T-cell related gene expression and EMT related gene expression. We were surprised by that because T-cell infiltration is generally felt to be a good thing, whereas EMT, at least the biological process of EMT, is generally associated with aggressive tumors and therapeutic resistance. And to make a very long story short, using computational approaches and leveraging PDX models, it turns out that fibroblasts are probably the key source for this EMT-related gene expression, and it's probably not the biological process of EMT that we're seeing in a lot of these gene expression signatures from bulk RNA data from urothelial cancer. So because these correlate, we wanted to see what this might be telling us about response to immune checkpoint blockade. And there is a statistical interaction between CD8 and this EMT, or what we call now stromal signature, so that if you have high CD8 expression, if your stromal signature is high, then you do worse than if it's low. And the same thing is true with response rate, progression-free survival, overall survival. In this statistical interaction reminded us of a spatial interaction that's been described with T cells and stromal cells. That is, it's probably simplistic to think about cold and hot tumors alone, but there's this intermediate phenotype that's been very well described where T cells actually get into the tumor, but they're restricted at the boundary, and they're restricted to stromal regions. Why this is associated with resistance to immune checkpoint blockade isn't entirely clear. My colleague Helen Salmon in model systems has beautifully shown that the extracellular matrix produced by cancer-associated fibroblasts probably causes a physical barrier to T-cell infiltration. The Roche group, along with Tom Powles and others, have shown that perhaps it's cytokines related to cancer-associated fibroblasts that are having an immunosuppressive effect. It's probably both, and there's probably more to this story here. What is important, though, is in the Roche study, this actually accounted for a huge proportion of the tumors. 47% of the tumors showed this excluded infiltrate. So this is probably not a minor mechanism of resistance to immune checkpoint blockade, but rather a major mechanism. So this picture is evolving now, where we have at least 3 independent predictive biomarkers associated with immune checkpoint blockade sensitivity. This inflammatory T-cell infiltration, PD-L1 expressing signature associated with response, mutational burden associated with response, and stromal or TGF-beta related signatures associated with resistance. How we might use these independent predictive biomarkers in the clinic is going to be challenging to analytically validate these and then implement them in clinical trials to define various subgroups based on the expression of these 3 different markers. But at least from a biology standpoint, I think this is telling us a lot about how we might move forward with combinations, as certainly there are combinations that are focused on overcoming stromal-mediated immune resistance, combinations with neoantigen vaccination in tumors that have a lower mutational burden, et cetera. So I'm going to stop there. Happy to answer questions.
So thank you for this great and wonderful talk. Are there questions from the audience?